Hindi nasusulat yung gilid. Bundled, ito yung tinatawag na chromatin. Okay? Medyo nakakalito yung image. Baka akalain nyo na ang chromatin ay nagiging chromosome. Chromosome, nagiging chromatin, ha? Not the other way around. Nucleoli are diffuse bodies with no surrounding membrane and are found within the nucleus. They consist of RNA and ribosomal RNA, and these are the sites of ribosomal RNA synthesis and ribosomal subunit synthesis. So nucleoli, ano meron sa kanya? Found within the nucleus, and they are the sites of ribosomal RNA synthesis, ribosomal subunit assembly. Okay, subunits will come into play later. Ay, sorry, I did not hit record. Ay, nagre-record na. Okay, sige. Good job. Okay, so next we have our nucleolus. The nucleolus is actually found inside the nucleus. Kumbaga, kung nucleus yung nasa gitna ng cell, the nucleolus is within the nucleus. Okay. Next, we have our ribosomes. These are organelles wherein proteins are produced. The protein builders or the protein synthesizers of the cell. They may be attached to other organelles such as the ER or our endoplasmic reticulum. And when it is not bound to any organelle, it is called free ribosomes. So dalawang ating type, bound and free. Kapag bound, nakadikit siya sa endoplasmic reticulum. Kapag free, wala. Very simple. Okay, so we have membrane-bound ribosomes. These are the color green specks you can see on the screen. And this is the microscopic view of our ribosomes. Did Ma'am Tricia explain to you what these images are? These images are actually very strongly magnified or magnifications of our cell. As a medtech, kayo, hindi kami. As a medtech in the future, you will rarely see magnifications of this strength. So ang ginagamit po ng medtech and general allied health professions is your compound microscope. To get these images, you actually need a stronger microscope, which is your electron microscope, your scanning electron microscope. So it is very hard to obtain these images. Yeah. Usually sa libro lang natin sila nakikita. So these are the differences of the ribosomes. The ribosomes here are the specks of black highlighted in blue on the right side of the screen. Free is kapag wala siya sa ating endoplasmic reticulum, and membrane bound kapag kadikit siya ng ating endoplasmic reticulum. Okay? Yung ating ER or yung ating organelle na nakadikit, yun po yung parang line. Okay? So ating free ribosomes, they tend to float around in the cell as in the diagram. Okay, so how is protein produced? Protein is produced by these steps. Also, there is a link, hindi naman link, there is a video embedded into our LMS. Please take time to watch it. It may be simple, but it is very intuitive to learn. Ribosomal proteins produced in the cytoplasm are transported through the nuclear pores in the nucleus. Papasok siya sa ating nucleolus. RNA, which is produced in the nucleolus, is assembled with ribosomal proteins to form ribosomal subunits. Yung ating ribosomal subunits, think of them as parang tagabuo. Siya yung tagabuo ng protein. The large and small ribosomal subunits leave the nucleus and the nucleus through the nuclear pores. The small and large subunits now in the cytoplasm combine with each other and with mRNA during protein synthesis. Okay? So ating ribosome, proteins, nagiging ribosomal subunit. The ribosomal subunit combines with RNA and forms proteins. Let's draw it para madali. Tama ba? Okay. Oh, nangyayari. Our ribosomal proteins form our ribosomal subunits. Okay? Kumbaga itong tagabuo. Okay. When the ribosomal subunits combine with mRNA, think of mRNA as the instructions. When the two come together, they form proteins. Naintindihan ba? So this, lahat ito nanggagaling sa loob ng nucleus. Nucleus. Klaro po?
Yes po.
Okay.
Po. Sir.
Yes po, sir.
Okay, next we have our endoplasmic reticulum, or our ER. It is a series of membranes forming sacs and tubules that extend from the outer nuclear membrane into the cytoplasm. We have two types of endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Our smooth endoplasmic reticulum is usually the site of our lipid synthesis, detoxification of chemicals within cells. Rough ER is the synthesis site for ribosomes. As it is studied with ribosomes and is the synthesis for protein. Okay? So para madaling may image pa sunod. Okay. So you can see in the next slide, the rough endoplasmic reticulum looks like this. Para siyang naka-fold. While the smooth endoplasmic reticulum looks like a sponge. Okay? Now, para madaling tandaan, smooth, fat, pag mataba ka, smooth yung balat. So, sa smooth, merong fat or fat ang sinisynthesize. Pag ikaw ay muscular, marami kang proteins, may texture yung iyong balat, rough yung skin mo, sa rough, nagagawa ang ating proteins. Okay? Saan po merong ribosomes? Ang nagpapa-rough ay ribosomes. Kapag may maliliit na bilog at hinawakan mo, rough yung surface, kaya siya rough endoplasmic reticulum. Okay? So this is the image of the electronic microscope image of the endoplasmic reticulum. And comparison between the rough and the smooth reticulum. Okay? So kita nyo another look at our endoplasmic reticulum. The rough endoplasmic reticulum is studded with ribosomes. While our smooth, no ribosomes. Okay? Paulit-ulit, pero malamang itatanong pa rin yan sa inyo. Next, we have our Golgi apparatus, also called your Golgi complex. If you're into internet memes, you should already know that hindi pala, mitochondria pala yan. Also known as the Golgi complex, the series of closely packed membrane sacs that collect, modify, package, and distribute proteins and lipids. Okay? Ang kanyang main role is to distribute proteins and lipids. Kaya nagpapack, modify, and package. Okay? Kumbaga, siya yung packaging center ng cell. Next, we have our secretory vessels. Secretory vessels come from our Golgi apparatus, in which kung ang Golgi apparatus ay yung packaging company, ang ating secretory vessels, siya yung transport van. Ang main role ng ating secretory vessels is to carry substances from the Golgi apparatus towards the cell membrane where the vesicle contents are released. It pinches off from the Golgi complex and moves to the cell membrane. So since Ma'am Trish already discussed your exocytosis and endocytosis, and it looks like this. Okay, so this is the Golgi complex. It is adjacent to your nucleus and endoplasmic reticulum. Okay? Kumbaga, dito ginagawa yung protein. Yes. Dito ginagawa yung mga requirements for the protein. Sa endoplasmic reticulum ginagawa ang protein. Then, pinapak siya sa ating Golgi complex. Okay, we have two phases of the Golgi complex. Para madaling tandaan, when cis ang pinag-uusapan, this is facing the nucleus. Kapag trans, it is facing away from the nucleus or farther away from the nucleus. So this is how the Golgi complex produces secretory vesicles and how it is used to release its contents. Parang nagkakameron ka ng bukol dito sa isang side, then it eventually will pinch off. Then paglabas ng pag lumapat na siya sa dulo ng cell, lalabas na yung ating contents. Okay? Alam niyo class na pinaka nakakairitang part na to? Bilang isang medical professional, most of the time na nakikita mo yan, isa lang yung sabihin yan. Magkakameron ng inflammation. Okay? Pag ang cells nag-release ng cytokines, example, na-detect ng cell na may damage around the surrounding area, it will release cytokines. Yung cytokines na yun, chemicals yun. Pag na-detect ng other cells, it will also release other cytokines and eventually mamaga yung isang area. Okay. Next, we have our lysosomes. So the main role of lysosomes is to destroy foreign materials. Is to destroy foreign materials. So it comes from the word lys, or to dissolve. Okay. So these are special organelles in which they contain strong enzymes that can break down phagocytized material or destroy foreign materials. So white blood cells carry a lot of these organelles. Peroxisomes, on the other hand, instead of destroying foreign materials, it carries enzymes that acts on chemical substances, such as fatty acids, amino acids, and particularly hydrogen peroxide. Hydrogen peroxide is a very reactive chemical compound that can destroy cells when poorly handled by the body. Okay? Kita nyo naman, pag nasugatan kayo at nilagyan nyo ng hydrogen peroxide na bula, it releases oxygen upon contact with organic material. The same happens inside the cell. If pinabayaan yung ang hydrogen peroxide to accumulate, it will eventually kill cells. That is why cells develop peroxisomes as a method to combat this problem. Okay? So lysosomes and peroxisomes are magkamukha. They look like small blobs around the inside our cytoplasm. Okay? So this is how our peroxisome or our lysosome acts. Kakainin ng cell, hindi naman kinakain. Kumbaga, magkakameron siya ng vesicle. Magkakameron ng vesicle formation. Papasok yung vesicle within the cell. Magre-release ngayon ng ating lysosome. This is a vesicle containing our compounds that can destroy foreign materials. Magdidikit yung dalawang vesicle. Mag-join together yung dalawa. Then eventually, papatayin lahat ng laman. Next, we have our mitochondria, the meme machine, or our power plant of the cells. These are small organelles with inner and outer membrane separated by a space. Other membranes have a smooth contour, but the inner membranes have numerous folds called cristae. The main role of bakit nagkakameron ng folds is to increase surface area. If you have a larger surface area, the more reactions that organ can accommodate. So the mitochondria are the major sites of production of ATP, which cells use as an energy source. The mitochondria can carry out aerobic respiration. When we say aerobic respiration, it means you consume oxygen. This requires oxygen to break down food molecules to produce ATP. This is your mitochondrion. Pag nakikita niyo yung folds sa gantong image, hindi siya actually tawag doon. Hindi siya talaga hindi naman talaga kita yung folds. Usually yan, cross section para lang makita niyo na may folds sa gitna. Okay? So the cristae of our mitochondrion ay itong mga folds na to. Kumbaga siya may mga hati-hati sa loob para lang lumaki ang ating surface area. Okay? So other images of our mitochondria. Mitochondria, we have the cristae. Pag nakita niyo itong matrix, fluid matrix. The fluid matrix is, kumbaga, yung tubig sa loob ng mitochondria. Okay, same lang po. Next, we have our cytoskeleton. Similar to the human body, which has a skeleton, the cytoskeleton is the skeleton of the cell. Primary roles of our cytoskeleton is to hold together the structure of the cell. Okay, it's the cell's framework. It is made out of proteins, and its primary function is to support the shape of the cell. It holds organelles in place, and it enables the cell to change shape. We have three types of skeletons, which cytoskeletons, which is our microtubules, microfilaments, and intermediate filaments. Okay. So the microtubules are the largest in diameter, pinakamalaki. The main role is to support cytoplasm of the cells. Support the cytoplasm of the cells, assist in cell division, and form essential components of cilia and flagella. Okay? Itong pangalawang to, this is one of the most important roles of the microtubules. In cell division, ang kanyang role is hatakin. Hatakin yung chromosomes na nag-divide. Hatakin yung kanyang chromosomes, magdadalwa into other poles. Para pag naghati yung cell, dala yung chromosome. Next, we have our microfilaments involved with cell movement. And we have intermediate filaments. So from smallest to largest, we have our microfilament. Then you have your intermediate. Then you have your microtubule. Para madaling tandaan, filament, intermediate filament, tubule. Yan ang tandaan yung words. The main role of our intermediate filament is to provide mechanical support to the cell. Okay, so this is the structure of our cytoskeleton. We have our microfilaments which surround the cell, intermediate filaments, and microtubules. Since microtubules are the largest, makikita nyo, mas thick yung kanilang lines. So this is another electron microscopy of your cytoskeleton. Okay, again, images. Tingnan nyo lang po. The slides are uploaded in Moodle. Okay, images ulit. Again, images. Okay, next, we have our centrioles. The centrioles are located in the centrosome and are made out of microtubules. It forms spindle fibers to separate chromosomes during cell division. They facilitate chromosome movement during cell division. Okay, so centrioles. Wait lang po. Cheng. Okay, so ang ating centrioles, they are found in the centrosome. It is a very small region in the cytoplasm, adjacent to the nucleus. Katabi siya lang ng nucleus. And the main role of the centrosome is to organize your microtubules. Hence, it is called a microtubule organizing center. Next, we have cilia, not the chair, but it projects from the cell surface. They vary in number and are capable of movement. It can cause movement over can move substances over certain cells. Okay, when we move on to our respiratory tract, cilia din po ang tawag dun ha. Yung mga ating hair within our noses, ang tawag din po dun ay cilia. Pero yung cilia na yun are comparatively very large compared to the cilia in your cells. Next, we have our flagella. Cilia is for movement of substances, meaning it does not move the cell. It moves things beside the cell. Kumbaga kaya niyang ilipat yung katabi niya, pero hindi niya kayang igalaw ang kanyang sarili. Unlike our flagella, it is much longer than this. It is much longer and stronger than our cilia. It is if you're going to compare it, it is it looks like a large tail. It usually occurs one per cell, and usually it is found in sperm. And its main role is to propel sperm cells. So this is your cilia. This is a normal cell. That is why it has no flagella. Okay, only sperm cells has flagella. Okay, so these are the cilia. Maliliit na protrusions from the cell. Okay, and this is your flagella in your sperm. Next, we have your microvilli. Microvilli are they look like cilia but are much smaller. Kaya po microvilli. They cannot move the cell, and they are primarily there to increase surface area. Okay? Increase surface area to increase absorption. Okay? These are primarily found in the GI tract, especially in your large intestine. Hindi siya for movement ha. It's not for movement. So sobrang liit niya, hindi niya kaya ibuin ng cell. Okay? It increases surface area. Okay? So, if ever, kung meron po may balak sa inyo mag-shift sa pharmacy, magiging importante po ito in the future. Kung balak niya lang naman. Okay, so this is your microvilli. As you can see, if this is your cilia, the microvilli is much smaller. Okay, this is what the microvilli looks like when cross-section and in SEM. This is the cross-section of your microvilli again. Okay. So, ano bang role ng isang cell? Generally, ang trabaho ng isang cell, it depends on what type of protein it produces. Okay? So, if your cell is in your GI tract, the proteins it releases primarily involves proteins or enzymes related to digestion. If your cell is located within the skin, the main role is to produce protein or enzymes that maintain the skin. So understanding how genetic information is used in the cell and distributed to daughter cells is important for understanding basic cellular activity. Okay? Now, maligaw lang tayo saglit. Have you ever heard of stem cells? Narinig niyo na po sa ating stem
Yes,
cells?
sir.
Yes
Okay.
po.
So why
Yes,
is
sir.
stem cells very kumbaga, a good field of research and a kumbaga, magandang treatment? Stem cells are undifferentiated cells. Meaning wala siyang exact role for now. Okay? Since lahat tayo nung bata or bago pa tayo maging fetus, we already have stem cells. These stem cells rapidly divide and differentiate. When you have stem cells, undifferentiated cells, you may place them in organs that need more cells. Okay? Take for example, one critical field of research is narinig niyo na po yung kasabihan na kapag na-damage ang neurons niyo sa utak ay wala na siya forever. Yes, that's partly true because neurons actually, the cells in your brain take a very long time to divide. Okay? Neurons, matagal mag-divide, pero neuronal connections, mabilis lang, maputol at mabuo. Pero yung neurons themselves, mahirap ma-recover. So in any case na na-damage yung brain or any critical part of the body, you may place stem cells in them and the stem cells will adapt to the cells already present in that area. So you may recover parts of the brain, you may recover parts of the liver, and kahit anong organs ninyo. Now, bakit siya mahirap i-research? It's because of ethical reasons. Since stem cells, katulad ng sinabi ko, these are undifferentiated cells, particularly from very, very, very young humans. One key source of stem cells is either the placenta of the baby itself, and your umbilical cord. Okay? So kung ako sa inyo at nagkaanak kayo ng bata in the future, you may want to preserve the umbilical cord and the placenta. Pwede po yung store actually. There are companies in the Philippines that will cryo your umbilical cord and placenta bago mapapakinabangan nyo siya 30-40 years in the future, if ever lang. Okay? Ethics aside, it's a very good field of research. However, pag kinonsider natin ethics, kaya mo bang kunan or gamitin ang katawan ng unborn baby for your research? Okay, going back, nahaligaw tayo. Next, so katulad na sabi natin kanina, the main role of the cell is to produce proteins for related to its function. So how does it produce proteins? Okay, so first we have our DNA or our deoxyribonucleic acid. It is a very long molecule that contains our unique genetic code. Like a recipe book, it holds all the instructions for making all the proteins in our bodies. It contains 4 basic building blocks or bases. We have our adenine, cytosine, guanine, and thymine. ACGT. The order and sequence of these bases determine the information available for building and maintaining an organism. Please memorize these 4 base pairs ha. I mean, yung 4 basic building blocks natin. And sino magka-pair? ATGC. ATGC. Okay? So DNA bases pair up with each other. A with T, and C with G. They form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule, particularly ribose. Together, a base, a sugar, and a phosphate are called a nucleotide. Nucleotides form together to form nucleotide strands, and these strands have a unique double helix shape, like a twisted ladder. Okay? Ito pa itsura niya. Ito mismo. The base pairs are forming the ladder's steps, and yung tabi are made out of your sugar phosphate backbone. Yung base pair, imagine yun yung steps bago yung mismong hawakan or yung handle. These are your sugar phosphate backbone. Each DNA molecule contains many different genes. So you may hear this a lot. When we say gene, it is a sequence of nucleotides that provides a chemical set of instructions for making a specific protein. So itong gene na ito, it is a sequence that uh, that produces a certain protein. DNA can replicate or make copies of themselves, which is a very important part of the process. So the ultimate expression of a gene is the production of a protein from the information contained in the DNA. So, depende kung ano nakasulat sa inyong DNA, anong lalabas na gene, anong lalabas na protein. Yung lalabas na protein, it will express itself as the phenotype we observe. When we say phenotype, magsa-cytogenetics naman kayo in the future. So, in short, phenotype is a physical characteristic. It is a characteristic. Take, for example, blood type. Blood type depends on your genes. The information encoded in a gene goes through a multi-step process, which is copied, transported, read, and finally used as a template for building proteins. We have different steps. We have transcription and translation, converting that copied information to a protein. Okay, now, paano siya ginagawa? Wala bang image dito? Okay, so ito yung summary executive summary ito. DNA. DNA will be copied. It will be copied via transcription. Okay. However, in this process, hindi saktong-sakto yung kopya. Kumbaga, i-convert niya into something that your ribosomal units can use. Kasi pag DNA, nilabas mo sa ribosome, hindi niya magagamit yon. Ribosomes can only take mRNA. So DNA to mRNA, the process is called transcription. Hindi siya kung naisip nyo, hindi siya duplication. Hindi siya copying. It's transcription. Writing something the same but in a different manner. Okay? Tinatranscript yung DNA strand into the mRNA strand. That mRNA strand will go out of the nucleus, dadalhin niya sa ating ribosomal subunits present outside the nucleus. Ang gagawin naman na sunod is babasahin ng combination ng inyong ribosomal subunits at ng iyong transport tRNA together with appropriate amino acid pool. Yung tinanscript kanina, babasahin, and this is called translation. And ang mangyayari po dyan, habang binabasa, kumukuha kayo ng certain amino acid. Then, depende sa pagkakasunod-sunod doon sa mRNA, depende rin kung anong pagkakasunod-sunod ng proteins. Okay? So, dyan po nabubuo ang ating proteins. Remember, amino acids become protein. Okay? As to what specific type of protein it forms, it depends on the combination and arrangement of amino acids. Okay, so in text form, transcription is the first step in gene expression. It takes place in the nucleus. It is a process in which DNA is copied to mRNA. However, it is transcribed, meaning hindi one-to-one or same yung pagkakakopya. Okay, ganito po yung nangyayari. Next, we have translation. It is the synthesis of proteins based off the information of the mRNA. The mRNA is found in transcription, is transported out of the nucleus into the cytoplasm, into the ribosomes. For a while, class, kuha lang po ako ng tubig. Okay, going back. Sa tayo. So this is the whole translation process. The mRNA is read by your ribosomal subunits. Your tRNA, which is the main role of the tRNA, is to attach or hold the amino acids in place. Sa video later na makikita nyo, naglalakad to. Okay? It actually reads 3 base pairs. At a time. These 3 base pairs are called codons. Okay? So every 3 base pairs or every 3 base pairs is a parang yan yung binabasa ng tRNA para alam kung anong tamang ikinakabit. So ang nangyayari po dyan, parang napasok at nalabas yung ating tRNA, nare-reuse yung tRNA. Okay? And step by step, humahaba ng humahaba yung chain. Okay? Pag tinignan nyo dito, mahirap basahin or mahirap maintindihan. It's much better if nagalaw. So that is why I put a video in the LMS for you to watch. Okay? Panoorin niya. Ito po siya. Okay. Haba nito Pang biology eh. Okay, and finally we have our cell life cycle. Okay? We have 2 major phases of the cell life. Dalawa lang ang part ng buhay niya. Dumadami, hindi dumadami. Kapag interphase, it is the phase within in between cell division. Okay? Meaning wala siyang ginagawa except mag-produce ng protein. Kapag cell division it is when the cells start to divide and multiply. Okay? So what happens during interphase? During interphase, nagpapadami tayo ng DNA. Okay? So interphase, nagpapadami tayo ng DNA. It is called DNA replication. Okay? It is slightly similar in a way to protein synthesis. But this time, instead of making mRNA, we make a copy of DNA. So the DNA in your chromosomes is replicated. 2 strands of DNA separate from each other, and each strand serves as a template for production of a new strand of DNA. Nucleotides in the DNA of each template pair Strand pair with nucleotides that are subsequently joined by enzymes to form a new strand of DNA. Kumbaga, one strand of DNA forms two strands of DNA. The sequence of nucleotides in the DNA template determines the sequence of the nucleotides in the new strand of DNA because adenine pairs with thymine and cytosine pairs with guanine. The two new strands of DNA combined with the two template strands to form double strands of DNA. In the end, a cell has two complete cells of genetic material, and the DNA is dispersed throughout the nucleus as thin threads called chromatin. Thin threads called chromatin. Thin threads called chromatin. Bubulol. Next. So cell division. We have two major types of cell division. Ang tawag natin ay daughter cells kapag kumbaga sila yung second generation. From single parent cells. We have mitosis and meiosis. When we say mitosis, kopya literal na one nagiging dalawa. As in literal copy. Meiosis, think of it as isa nahahati into two. So meiosis this is a type of cell division reserved for sex cells. So each cell of the body except for sex cells contains 46 chromosomes. Sex cells have half the number of chromosomes, particularly 23. 46, the diploid number, are organized to form 23 pairs of chromosomes. Okay? Wag malito. 46 ang chromosomes, 23 ang pairs. Okay, we divide them into pairs. Of the 23 pairs, one pair is the sex chromosome. One pair is the sex chromosome. It determines the gender. So it's either XX or XY. XX is the female and XY is the male. So usually yun yung the only difference between the 23 pairs. The remaining pairs of chromosomes are called autosomes. The sex chromosomes determines the individual sex, and the autosomes determine most of the characteristics. Okay? In relation to these 23 pairs, if a certain disease is linked with the sex chromosome or yung hindi autosome, dyan po lalabas yung mga conditions na sa lalaki lang lumalabas. Okay? So, in particular, if the disease only appears on people with Y chromosomes, meaning lalaki lang, sa lalaki lang lalabas ng disease na to. A good example is color blindness. A certain type of sex-based color blindness. Okay? Kaya kung mapapansin nyo, most of the colorblind people are male. Okay? And kapag marino kayo, malamang lalaki ka. Kailangan mong Ishihara color blindness test to determine if you can see colors. Hindi naman see colors, kumbaga ay detect the correct colors. Okay? Also male pattern baldness. Mitosis occurs in all cells except sex cells. A parent cell divides to form two daughter cells with the same amount and type of DNA as the parent cell. Divided into four stages, we have prophase, metaphase, anaphase, and telophase. Okay? Para madaling tandaan ito ating acronym PMAT. Guys, ang pa magsulat. Ang ating acronym ay PMAT. Prophase, metaphase, anaphase, telophase. Magkakaibang letters. So madali lang siyang tandaan. Okay? So this is our summary ng ating cell division. May DNA ka, padadamihin mo ang DNA. Pag doble ng DNA mo, magmamitosis ka, Yung DNA, yung isang half ng DNA pupunta dito, yung isang half ng DNA pupunta dito. Mapuputol yung dalawang cells and each will have its own set of DNA. Hindi ko alam kung bakit baligtad to. Okay? Okay, now DNA hindi naman siya hinahatak onto opposite sides of the cell as DNA. DNA is packaged into chromosomes first. Then chromosomes through chromatin, kumbaga babalot muna siya sa protein para maging chromatin. Chromatin magfo-fold, magiging chromosome. Okay? So ganito ang itsura ng ating chromosome. Okay? Kaya ang maganda po dyan, kita nyo parang symmetrical siya. Kita nyo yan, symmetrical yan. Mamaya pag hinatak yan, itong side na pupunta dito at itong side na to pupunta dito. Okay? Nahahatak siya in between. So you have your DNA replication. Kapag pag nagdoble ng DNA mo, it will condense into your sister chromatids. Okay. So ito yung itsura, kumbaga ito yung itsura ng chromosome na ready ng hatakin. You have your centromere here. Kaya centromere, it is in the center. It binds the two chromatids. Ang isang chromatid, itong isang to. Isang mahabang to, chromatid to. Isang mahabang to, chromatid din to. Okay? Next, we have itong kinetochore. Itong kinetochore, dyan na parang, kumbaga ay hawakan. Hawakan or a place of attachment. Yung kinetochore, pinagkakabitan ng ating microtubules. Ang anong role ng ating microtubules? The role of our microtubules is to pull on our chromosome to pull them apart. So chromatid, these are two genetically identical strands of chromatin. A centromere is a region of chromosome that joins two sister chromatids. A centriole, it forms the spindle fibers to separate chromosomes during cell division. Kinetochore is a specialized region of centromeres of chromosomes, and cytokinesis is the process of division of the cell, bringing about the separation of two daughter cells. Okay? So ito po ang ating phase ng ating okay, ito yun eh. Ito yung mismong cell cycle. Pero nakafocus lang siya sa mitosis. Okay? So prophase, metaphase, anaphase, telophase, and cytokinesis. Cytokinesis is the last part in which totally nagsaseparate ang ating ating cells. Okay. So let us describe each part of the cell division. Each part of cell division is marked naman with kung ano nangyayari. Okay? So, kung tayo nasa classroom ngayon, one of our activities should have been gagayat kayo ng onion. Kung kayong onion, ilagay nyo sa microscope bago hahanapin nyo kung aling cells ang nasa isang part ng cell division. Ang nga pala class, please take note that not all cells are divide at the same time. Okay? Hindi siya, kumbaga yung layer ng skin mo, hindi lahat ng cells pare-pareho ng phase ng cell division. Okay? Irregular yan. Okay? And not all cells in the human body replicate at the same time. Kaya kung napansin nyo, bakit mabilis tumubo ang buhok compared sa nails, okay ba yung rate of cell division niya? Okay. So we have prophase, the first process, prophase. Chromosomes condense and becomes visible. Nawawala ang nuclear envelope and the nucleolus. Spindle fiber apparatus forms. Okay? So nagsasama-sama po sa gitna ng cell ang ating chromosomes. Okay? Then nagsisimula na pong lumabas ang ating microtubules. Itong itsura ng microtubules para siyang thread na humahaba. Okay. Late prophase, gumigitna sa center plate ang ating chromosomes. And nagsisimula ng lumapit ang ating spindle fibers. Metaphase, naka-align na ang ating chromosomes. Kanina nagko-condense lang. Pero ngayon naka-align na within the equator. Kumakabit na ngayon ang ating Kumakabit na ngayon ang ating spindle fibers. And that is our metaphase. Anaphase, hinahatak na ngayon ng spindle fibers yung ating chromosomes, separating them from each other. However, please take note, among the equatorial plane, hindi pa naghihiwalay totally yung cells. Nagsisimula pa lang pero hindi pa naghihiwalay or nagsasara. And finally, we have our telophase. Telophase, nagko-condense na yung nagko-condense at nag-unravel na ulit ang ating chromosomes. It will start to reform the nucleus on each side of each new cell and nagsisimula ng humati yung original cell. Okay? So this is the summary of our mitosis. Okay? On the left side, you have your representation. On the right side, you have your actual cell division. Ito, naobserbahan to sa microscope natin. Kaya siyang tingnan ng ating microscope. Okay? So, PMAT. Cytokinesis. Okay? Sorry, I can't play this video for some reason. And finally, we have our cell death. Okay. Now, apoptosis is programmed cell death. Why do cells need to die? Cells need to die because DNA that has been replicated for multiple times can lead to errors. Okay, if you recall, okay, yung ating DNA, di ba, double strand siya. One DNA. Okay, bago manganganak siya. Okay, into DNA ulit. 1, 2. Pagpalagyan itong ating isang strand, dalawang double-stranded DNA, ito pupunta dito, ito pupunta dito. Okay, then makokopya siya, may bagong strand na magpo-form. New. This process is called semi-conservative. What does it mean? It means along the lines, yung DNA sa pinakaunang cell ay dala pa rin along the lines. Hindi nawawala yung lumang DNA. Okay? Kahit pa pag-replicate ulit yan sa generation na to, it will still carry the DNA. One of them will still carry DNA from the first part. Now, what is bad about this? DNA that has been replicated for too long may lead to errors in encoding, errors in replication, and those errors might lead into something more malicious like cancer or wrong protein formation or any disease. That is why normally cells should die. So we have apoptosis. Apoptosis is normal cell death. It is a normal process in which the cell numbers of various tissues are adjusted and controlled. Hindi naman pwedeng dadami ang iyong cells nang hindi nababawasan. Pag nangyari yon, isa kang magiging living cancer. In As apoptosis begins, the chromatin within the nucleus condenses and fragments, and fragmentation of the nucleus equals cells and fragmentation of the cell. Okay? Kumbaga, yung DNA nagiging unusable na. Specialized cells called macrophages phagocytize the cell fragments. We have different factors affecting aging of the cell. Cellular clock, death genes, DNA damage. DNA damage can result from errors or radiation. Free radicals, like our highly active radicals, free oxygen, and mitochondrial damage. Okay. So in essence, if the cell does not have a mechanism to kill itself, it will become cancerous. Okay. Cells that fail to stop themselves from dividing become cancers. Okay. So that is it for our lecture. My final instructions to you for today is yung mga hindi pa po nag-attendance, mag-attendance na. Then please make sure you watch the videos posted in LMS. Okay? And again, an exercise will be uploaded within the week. Pasahan po natin ay Thursday ng hapon. Once the exercise is uploaded, I will ping the group or I will ping the team again. And may quiz ulit po tayo on Friday. Are we clear?
Yes po.
Yes po.
Sir.
Po.
Yes
Okay.
po.
Sir.
So, yes
Sir,
po.
ano
sir.
pong i-quiz? Yung chapter 3 pong buo o kasama po yung i-discuss nyo pang chapter 4?
Ang alam ko ay kasama ang chapter 4 na
Sir,
ang
same
alam ko
time
nga.
po ba?
Oo. Wait. I cannot assure the same time. Baka mag-iba na naman ang isip ng leader. Pero I can assure you na sabay-sabay kayong kukuha. Okay, baka
Sir,
baka mga alas 6 ulit. Okay? Any more questions? Magkikita pa
Sir,
naman tayo ng
excuse
Wednesday.
po.
Yes po.
Sir, excuse po. Iba po ba yung group na nasa lec tsaka dito sa atin sa lab?
Hindi dapat. Bakit? Magkaiba ba?
I mean, iba po yung group na gagawin nyo? Or
Wala
yung
pa-
pa rin po?
Wala pa ba tayong group? Parang may group na tayo ah.
Same lang din po dun sa Anafi, like, okay. Sige
Bakit?
pa.
Magkaiba ba?
Hindi po, na-confuse lang po ako.
Okay, sige. Pero ano lang, magka-mirror dapat yan. Unless may sumaling bago in which, well, sa last group siya papatak. Okay?
sir, question po.
Sure, ano po yon?
Yung activity po by group ulit.
Yes po.
Okay po, thank you po.
Okay. Ano pa po? May question pa po kayo?
Sir, same
Yes.
leader pa rin po?
Yes. Actually, wala namang purpose na sinaserve yung leader except para maging pula yung unang name.
Sige po.
Any more? So please turn on your webcams so I can take a screenshot para may ipasa ko for my attendance today. Thank you. Okay, so I will be taking it now. And 1, 2, 3. Okay. So thank you class for participating. Let's meet each other again on Wednesday. Make sure to read the PowerPoints together with watching the videos. Okay, thank you. You are now dismissed.
Thank you po.
Thank you, sir.
Thank you po, sir.
Thank you po.
Thank
Thank you
you.
po.